Dr. Mario here, and today's topic is source to sink movement in plants. Photosynthesis is the process by which plants convert sunlight energy into sugars or glucose using carbon dioxide and water while releasing oxygen to the environment as a byproduct. For carbon dioxide to enter the plant, the stomata of the plant needs to be open, and the environment exerts a great deal of influence on opening and closing stomata. Stomata opening depends on light intensity, temperature, vapor pressure deficit, and water availability. So stress mitigation and sound nutrition management are key for successful photosynthesis. Sugars stored in the leaves are distributed in the plant by the phloem tissues. High demand for sugars in the zinc tissues like the roots, the corn ears, or the soybean pods induces fast uploading of the sugars from the leaves to the phloem cells. So the source to sink relationships control the flux intensity in the phloem tissue. So the question is, how does the source to sink movement occur? Sugar flow gets directed and unloaded in the zinc tissue by the pressure flow model first proposed by Ernst Munch in 1930. In this model, sugars or sucrose from the source or the leaves, concentrates at the companion cell, and this draws water from the xylem to the phloem and creates some pressure to move the sugar from the companion cells to the phloem. As sugar moves down the phloem, the zinc companion cells pull the sugars into the zinc tissue, and as the concentration of sugar in the phloem near the zinc is reduced, water returns to the xylem and the process repeats again. So sugars move from high concentration to low concentration, and this loading and unloading of sugars require energy in the form of ATP. The movement intensity in the phloem could be potentialized by some nutrients like boron and potassium. Potassium is the element responsible to control the osmotic potential in the phloem, and boron makes sugars transport more fluid. In addition, the presence of other nutrients such as magnesium in the leaves facilitates the sucrose movement from source to sink by driving the ATPase pump. Zinc acts as a cofactor in the enzymes responsible for starch breakdown, and under zinc deficiency, high levels of starch are found in the leaves. In summary, to maximize photosynthesis and sugar movement, NEXTA Spark and NEXTA Swole can help by adding the right hormones and nutritional components when needed. Contact your local NEXTA biological representative for more details on NEXTA Spark and NEXTA Swole. Until next time, is Dr. Mario reminding you to stress less and yield more.